as well as also protecting biodiversity or marine biodiversity, of which the Philippines is known for. So the Philippines is the third largest producer of seaweeds in the world, equivalent to um, 4.5% of the world production of marine aquatic plants. And in terms of the cumulative annual world aquatic plant production, it is also clear that Asia places an important role in terms of producing the global seaweed production, which has already exceeded in about 30 million tons per year, almost all of which come from aquaculture sources. And based on these figures shared by the Food and Agriculture Organization, production has
Engineer?
tripled.
Yeah, I'm ready.
I'm sorry. Um, Production has tripled between the year 2000 and 2016 in continuous growth, with the average yearly incrementals of about 7%. So the Philippines is one of the among the top 20 seaweed producing countries in the world for the period 2010 to 2016. And this is the summary of the statistics that we gathered at the start of our study or research with the CFAS in UK. So, but we have also to note that as compared to the other countries in the top 20 producing um, um, countries in the world, our percentage per year has been annually decreasing for as much as 22%, negative 22%. So this made us ask the question as to what were really the factors, if we are supposed to be in the top 20 producing countries, why are our production decreasing from that year 2010 to 2016? And in terms of the importance of aquaculture production in the country from marine waters, it is evident that seaweed accounts by weight for most of the production, which is equivalent to about 1.4 million tons wet weight. However, when considering the value of aquaculture products, seaweed accounts only for 9% of the total value. So although they have the largest in terms of production, in other words, they, in terms of translating to economic benefits, they only account for a measly, less than 10% of the value. This is according to the FAO statistics. Um, in terms of the seaweed production from aquaculture, this is a slide which shows to us what were the predominant species that were used in the Philippines in terms of aquaculture. So we are predominantly using the Kappa-Picus alvazeri, which is a source of Kappa carrageenan, or the hard gelling colloid, and Lambda carrageenan, or the non-gelling colloid. So other species that we are using also are Qualerpa, Gracilaria, and Eucheuma. In terms of the seaweed production from aquaculture by species, it shows to us that predominantly we have, based on this figure, the Kappa-Picus is also producing the majority of our production from the 1980s until the 2016 period. And so the focus is also on Eucheuma in some of the areas. So there are species that have produced in terms of seaweed production are really the Kappa-Picus species. So the rationale of our study was that, we have seaweeds that can be divided into three groups based on their pigmentation: red, brown, and green. They have been harvested and used by humans for millennia as food sources and for medicine. And although we have a lot of species, about 500 species of seaweed, only 33 genera are harvested and are farmed commercially worldwide. But as demand increases, production of seaweeds have moved from wild to farm sources. That's why we have seen a growth of seaweed farms, especially here in Central Philippines, where we are located. So majority of the seaweed production is for human consumption or for flavorings, while the remaining is for production of phycocolloids for animal feed, fertilizers, probiotics, and for water remediation. However, aquaculture sector in the Philippines is considered to contribute significantly to the country's food security and employment because it also provides or improves the socioeconomic status of coastal communities by generating employment to them or diversifying them from fishing livelihoods, which is their regular mode of earning, to aquaculture, seaweed aquaculture. It also enhances, as you can see later on, community cooperation, especially in island communities, and promotes small and medium enterprises. And of course, it actually results to more stewardship of the marine resources because these are the resources that they are most likely directly reliant upon. We have also seen from studies that aquaculture can actually help reduce pressure on wild fish resources, allowing the recovery of our overfished stocks already. But there are all other risks that we are seeing, one of which would be in the sensitivity of the habitats, and of course, the effects of climate change as well. So our objective, which is part of the Newton Institutional Link program, is to develop a toolkit or methodology to identify what would be the suitable seaweed aquaculture production sites, which maximizes economic returns while minimizing environmental effects. So we wanted to see a balance between economics as well as environmental protection. We focused our project on in the municipality of Bantayan in Cebu, Central Visayas, because we think that there were a lot, this is an area where there's not been much study as we were trying to look for data around the Philippines. We looked at sites where there have been very sparse information regarding seaweed or aquaculture production. And so we focus also in particular to the island barangays as shown in the picture or in the slide here. So these were once we went into these islands, we saw that these were the seaweed species that they were cultivating in Bantayan Island. So mostly were Capapicus, as can be seen. So they actually reflect the national data also on the amount of the number or the kind of species that they were cultivating. These were the farms that they were using in the area. So when we went there, these are the farms that you could see in Bantayan, the small islets around Bantayan Island as well. So their farms would be around 100 meters, square meters. So the post-harvesting process seems to be a very, community organized process as well. So here it's shown that they dry their seaweeds on a platform or by hanging it and after its subsequent removal of seaweeds from the rope. So you could see that most of the women were actually engaged, including their children, in the post-harvest stage of seaweed farming. So the activities actually were to review the status of seaweed aquaculture in the Philippines, seaweed farming techniques used, and provide a very good overview of the study area. We collated socioeconomic and environmental data. We went into the development of the multi-layer GIS toolkit to identify areas with the most suitable conditions for seaweed farming and also to reduce potential conflicts with other activities such as fishing, navigation, and also conflicts with sensitive habitats such as marine protected areas. And then we provided recommendation on optimal location of aquaculture facilities. So overall, we wanted that our recommendation would be done with active local stakeholder involvement to facilitate long-term sustainability of their aquaculture production and integration of these activities with other livelihood benefits that they have in the area. So we started our institutional link program by visiting our partner institution in the UK We were also shown the practices of using the GIS toolkit in seaweed seaweed aquaculture in the UK, and how this has been used to also provide recommendations for more increasing the productivity of these aquaculture farms. Afterwards, we went to the communities and engage our stakeholder communities to identify what would be their environmental and socioeconomic priorities. We gathered existing data on aquaculture facilities at our study locations. We gathered environmental data layers that we'd be using for the GIS Marine Spatial Planning tool. And we also gathered, of course, the socioeconomic data from by conducting interviews with all our stakeholder communities. So these were some of our activities as shown here. So we would go to the islands regularly and talk to the stakeholders and gather data from them. Afterwards, we decided to also, in order to facilitate our data collection, we developed an app, a web and mobile app for data collection, and which would automatically feed into our analysis of the information that we gathered. We also in the process, when we realized that women were essentially, there were gender issues that we wanted to focus or hone in on in this project, because of the presence of women in every stage of the aquaculture production. So we conducted also gender empowerment survey. So this was the multi-layered GIS toolkit that we were able to develop out of our data gathering. So this is the methodology used to create the spatial planning GIS tool. So we had to look looked at a human activity index. We also looked at the environmental index and the socio-economic index, and so to be able to determine the seaweed's suitability index in the area. So for this, we were able to classify environmental data, and shows also data in Bantayan Island, where the minimum and maximum percentile for low and high seasons were gathered based on the optimal temperature range required for the production of seaweed. So we could feed into the GIS toolkit information, the fields that we wanted to highlight and we wanted to improve on with this toolkit. So some of the key results.
Just the last minute, yes? A gentle reminder. Just 1 minute left. Thank you.
Yeah, okay. So some of the key results we have was we were able to classify our data and find the suitable areas for seaweed production. So this is some of our generated. So we also did capacity building of local stakeholders. We trained them on seaweed farm management and appreciation of coastal systems. We also wanted to share research skills with local universities in the area in Bantayan Island. And also, of course, we introduced to the stakeholders at the end the GIS toolkit for their consideration and adoption. And we also had livelihood training for food and non-food products using seaweeds in order to provide also the benefits. So this is the video that we left in order for them to understand what the GIS tool is all about. Finally, as I said, we wanted to look at the gender issue, and we found out that women were actually more active in farming tasks than men, but they had more decision making power or empowered if they had years of experience, as well as more income generated from seaweed farming. So key lesson would be long-term sustainability of aquaculture requires integration of information with other activities. A spatial planning tool allows for the combination of various spatial tools or data. So this is a flexible open source tool that we could generate information about, so that we can realize the trade-offs between environmental status and economic and social benefits. Thank you so much. We have several recommendations here. I would just like to show you because I don't have any more time. Okay, I would be happy to discuss this in our open forum. Thank you so much.
Thank you very much, Dr. Osorio. And yes, welcome our participants to share any questions in the chat, and we can have a Q&A session at the end. It's great to hear about your work on aquaculture and also all the GIS spatial layers that can integrate the information, and also about how it impacts those development goals of food security and employment. Thank you very much. At this time, I'd like to ask Dr. Romero to please speak to us. The floor is yours. Thank you.
Yes, hello everyone.
Hi.
Good afternoon.
Yeah, let me just share my screen. Is it okay now?
Yes, we can see. Thank you.
Okay. First of all, thank you, Newton Agham, for this opportunity to present our Newton project, Enrich. This is in collaboration with Rockham Said Research, Alison Lovegrove, Andre Kosick, Peter Shuri, Iri with the Nese Sriniva Sulu, and my colleagues here at PhilRise, Evelyn Bandunil and Risa Abilgos Ramos. Included in the SDG are good health and well-being and responsible consumption. And also in the world food systems, there's nutritious and healthy. So the challenge for the breeders is to consider either quantity or quality in terms of nutrition. Now, we all know that rice is a very important crop in the world because it is basically the staple food for half of the world's population, including the Philippines. But unfortunately, we consumed it in polished form, where the more nutritious bran and germ are already removed. So white rice or polished rice is just basically 90% starch with low amounts of protein, dietary fiber, and micronutrients. So it's our main source of energy, but unfortunately, it has a relatively high glycemic index. And as we know, the consumption of foods with high rapidly digested carbohydrates and low dietary fiber are associated with increased risk of a number of diseases. This is the typical Filipino diet, and as you can see, it's carbohydrates heavy with low amounts of fruits and vegetables. And of course, rice is the star of the meal, comprising about 34%. And we also know that eating too much rice will pose health risks for different diseases. Now, let's look at the leading causes of death in the world. And topping the list are heart disease, stroke, cancer, and diabetes. And these are all attributed to rapid urbanization, unhealthy diet, sedentary lifestyle, and change from our traditional crops to more processed foods. So basically, it boils down to unhealthy diet and physical inactivity. And if you look at the Philippine scenario, it's basically the same. It's heart disease, stroke, cancer, and diabetes. Now let's take a look at diabetes, which is a long-term metabolic disorder that is characterized by high blood sugar and insulin resistance. So this is really alarming because diabetes is on the rise. And you can see here in this figure the projected number in 2030, and they are all here in Asia. So in managing diabetes, we encourage high fiber, low glycemic index carbohydrates. So these are the whole grains, pulses, fruits, and vegetables. And there are already accepted health claims from both the EFSA and FDA. And here, dietary fiber is plant cell wall polysaccharides, and resistant starch is considered a dietary fiber. So we can see here that there's an increased increased dietary fiber content in our food. It slows the release of glucose. So you can see if we compare eating a high fiber bread versus an average fiber, that there's a rapid increase in the reducing sugars upon consumption of the lower fiber foods. And you can also hear the spike in the blood glucose levels with the consumption of high GI foods. So the objectives of our projects is to identify the differences in the content and composition of the resistant starch and dietary fiber, and to select lines with high amounts of these two components, determine also the grain quality, especially the palatability and the stability of the traits. And of course, the ultimate objective is to provide genotypes and markers to national breeding programs. So we did high throughput screening of the dietary fiber of 370 rice lines. So basically separating, converting the polymers into the monosaccharides. And you can see here that the majority of the monosaccharides from the cell walls were in the water unextractable fraction. And this is the principal component analysis, cluster analysis. And you can see these are the rice lines that have higher fiber, and these are the rice lines with the lower fiber. And then this is looking also at the more detailed characterization of the dietary fiber in the different fractions, and also the rice structure. The dietary fiber structure of the rice. And then you can see here the differences between the cereal cell wall components between wheat and rice. And then we also look at the resistant starch. So this is basically the work of Erie, because we know that if we increase the resistant starch as a dietary fiber in staple foods, it can counter the non-communicable diseases by reducing the risk factors, especially for diabetes. And these are the lines that were selected with relatively high resistant starch. And these lines were used in the multi-site trials and also in the sensory evaluation. And then they also did gene expression studies. So you can see here their characterization and the results of their gene expression work. The 9 lines were planted at field rice from 2017 wet season up to 2018 dry season and 2018 wet season. So these are the 9 lines, and we characterize the grain quality in terms of the crude protein, amylose, which is basically a component of starch, gelatinization temperature, which is an estimate of the cooking time of rice, and also the texture in terms of the cooked rice hardness. And then we also conducted sensory evaluation in terms of the percent acceptability, and the mean rating for the overall parameters, as well as the texture, and also in the cohesiveness, tenderness, and smoothness. So you can see here the different lines, and Filipinos really like the tender samples compared to the harder ones, and that's really a challenge for finding a low GI rice. And then we did some correlations between, for example, here, the acceptability and the amylose content. So you can see here, these are the low resistant starch and this is the high resistant starch. As the amylose content is increased, the acceptability is decreased. And the same thing, if you increase the amylose content, the texture is also increased. And these are the other correlations among the different grain quality parameters and also the sensory properties. And then we also conducted consumer survey where we asked the perception of rice with high resistant starch, dietary fiber, and low glycemic index. And the responses are really positive, especially in the nutritional value. So they really like this kind of rice if we ever will have this, because it really addresses our nutritional problem. So basically, we were able to screen a diverse rice genotypes for this resistant starch and dietary fiber, and we selected promising lines with high components of these two. And then these lines were used in both in the G/E studies and also in glycemic index screening, as well as in the grain quality, especially the palatability. And now we are recommending that lines with improved dietary fiber and resistant starch content with high amounts of this and also low GI with good palatability need to be pyramided so that it will be available later on with all these good components. And through additional funding, there were training exchanges. So this like from Rothamsted to Erie, and also included in the Starch University to Rothamsted. And we also had a chance to go to Rothamsted Research for short-term training for the dietary fiber analysis. And then we generated 3 publications out of this project. And so again, we would like to thank Newton Fund for this opportunity, and also the fund that we got from PhilRise as well as ERI as the counterpart fund. And again, thank you so much for this opportunity to share with you our project. Thank you very much.
Thank you very much. Thank you for your presentation and sharing. And it's so good to hear from Enrich, this project, and also on the research on rice and also on non-communicable diseases, as you mentioned. Let's see if we have some questions at the Q&A. Thank you. And last but not least, may I now invite Dr. Dennis Umali to speak, please. The floor is yours. Thank you.
Hi, Dr. Dennis, you might be on mute.
Good day, everyone. Magandang hapon sa ating lahat. Can you hear me now?
Yes, we can. Thank
Yes.
Okay. So first and foremost, we would like to thank the British Council and Dr. Danny Gonzalvo for the invitation to be a part of this very wonderful event. And on behalf of my collaborator, Dr. Muhammad Munir of Lancaster University, it is a great privilege to be here with you this afternoon. Dr. Munir and I were the recipient of the Newton Agham Researcher Links Workshop in 2018. Dr. Munir is a lecturer from the Lancaster University, and I am an associate professor at the College of Vet Med, UPLB. Our approved workshop links is entitled Novel Vaccines and Diagnostic Technologies Against Emerging and Re-emerging Veterinary Pathogens. And this was held last February 4 to 7, 2019 at Rizal Park Hotel, Manila, Philippines. And this was funded by the British Council and Newton Fund and the Department of Science and Technology through Secretary de la Peña, and the Livestock Research Division of PICARD through Dr. Ebora and Dr. Sainan Baguio. Now, for this workshop, the objectives were as follows. First is to forge long-term research partnerships between researchers in the United Kingdom and the Philippines for better design, delivery, and formulation of vaccines and diagnostic technologies to reduce the impact of viral diseases in livestock, poultry, and as well as human beings. Number 2, to organize a functional network of scientists and vaccine and diagnostic specialists covering major areas of animal viral diseases that will identify major research gaps and funding opportunities related to vaccines and diagnostics. Number 3, to establish a multidisciplinary platform for training of early career researchers regarding vaccine designs and diagnostics. And number 4, to identify funding opportunity from international, UK, and Philippine funding agencies for future studies on vaccine design and diagnostics. So here are some videos and clips during the workshop. Now, some highlights of our workshop. In total, it was around 4 days in length, divided into 11 sessions, wherein 8 were scientific sessions and 3 were interactive sessions. All in all, we invited 25 expert speakers from 11 academic institutions, 2 research institutes, 1 government agency from the United Kingdom in every parts of the Philippines. No? So some of the speakers were from the Purbright Institute, Brunel University, Lancaster, Royal Veterinary College, London, University of Liverpool, University of Nottingham, University of Surrey, UPLB, Central Luzon State University, Cavite State University, University of Eastern Philippines, Angeles University Foundation, from the Department of Agriculture, Bureau of Animal Industry, and the Philippine Carabao Center. Now, all in all, we received around more than 100 applicants for the workshop, but due to the limited resources through a selective screening committee composed of experts, we were able to narrow down the participants to 70. And these are some of the participants. No? And out of 70, 30 were senior mentors from the United Kingdom, and 57 were from the Philippines. And all in all, 13 universities, both from the UK and the Philippines, participated in this workshop, 4 research institutes, 10 government agencies, and 1 private institution, as well as 3 media partners, DOST TV, Alagang Magaling, and Pit Games Media. Now, I'll share some video. Just wait a moment.
So while Dr. Dennis is preparing the video, I'd like to share that the video he's about to share is a coverage of the Department of Science and Technology, DOST STII, when they managed to complete the workshop in Rizal Park Hotel.
Okay, thank you, Danny. And to add to that, no, this video highlight was made by Pit Games Media, the executive producer of Tucan, a poultry show being broadcasted in the Philippines, in Southeast Asia, and in the United States.
Welcome po sa Newton Agham Researcher Links Workshop. Ang Newton Agham Researcher Links Workshop ay isa sa mga programa ng British Council wherein ang mga expert sa United Kingdom at mga expert sa Pilipinas sa larangan ng bakuna at diagnostic techniques ay pag-meet natin para mag-exchange ng ideas, technologies at information in the hopes na makabuo ng bagong bakuna, diagnostic kits, o anumang tools na maaaring magamit ng ating mga farmers para mas mapagana nila o mapaunlad nila ang kanilang mga farmers. Sa simula, nung pinaplan na itong event na to, we were expecting 20 top-of-the-line experts from the UK and 20 top-of-the-line experts from the Philippines. Dahil sa tulong ng Picard, so mas dumagdag pa, no? Dagdagan ng funds, kaya lumaki. So from 40, ngayon po ay nasa 60 to 70 na ang ating delegates.
Ako po ay naimbitahan dito bilang speaker para mag-present ng emerging and re-emerging diseases of veterinary pathogens. Sa mga kasama ko po, ito po ay isang magandang venue para mag-sharing po tayo ng information, magandang venue po para magkaroon ng mga partnership, para mapag-aralan po natin kung ano yung mga sakit na kailangan po natin i-address sa ating industriya. Itong ating pong ginagawa ay para naman po ito sa ating mga livestock farmers, sa ating mga poultry farmers. Kapag nakadevelop po tayo ng mga diagnostic kits or di kaya mga bakuna po na kailangan natin sa pagsugpo ng mga sakit, ito po ay makakatulong para maitaas yung income, maganda yung production ng ating mga alaga, and then mas mapaparami natin sila.
Sabihin natin na ang event na ito ay parang blind date. So, dinidate natin ang mga taga-UK with the Philippines, introduce them, na ito ay expert niya, ito ay expertise niya, in the hopes na sila ay makabuo ng research proposal or any products na gusto nilang i-develop, na in turn, isa-submit nila sa British Council para pondohan. So, this is the first step of a long process na ang endpoint ay makadevelop ng bagong teknolohiya, particularly tungkol sa bakuna at sa diagnostic.
So this initiative is usually a collaborative option contributed by the British government and also by the Philippine. DOST is a Philippine organization that is contributing into it. And the primary aim of this Newton Agham Research Link workshop is to give an opportunity for people coming from other country in Philippines so that Filipino and, for example, in this case, British, they come together and identify the research gap that are required to be addressed in the future.
So it will help because we're collaborating with the university in Los Baños, and we're hoping that collaboration, as well as some of the collaborations that come from this, will help reduce these zoonotic pathogens and reduce human disease. Yeah, it's been really positive so far. Everyone's really helpful, everyone's really friendly, and everyone's really willing to come and speak to everybody else, so it's very good.
This event is really, really helpful, especially for interaction with the young people here, as well as the senior people, to long-term establish a collaboration between United Kingdom and Philippine scientists, and it will go a long way.
I think the fact that it's brought so many people together in not only a scientific event, but also a social event, so that we understand each other better. We understand the problems, we understand the work that we're doing, and we understand how to produce a solution. Is really important.
Kami po ay lubos puso nagpapasalamat sa DOST and sa British Council sa pagsuporta para ma-execute namin ang isang napaka-prestigious na event na to sa Pilipinas. This is the first time in the history ng PetNet na may ganito.
Okay, so that was the highlights of the workshop. In summary, there were 25 papers presented, mainly on the status and epidemiology of veterinary diseases in the Philippines, advanced topics on vaccine design on swine, poultry, and other livestock diseases, advanced topics on veterinary disease diagnostic, monitoring tools for antimicrobial resistance, diagnostic techniques for zoonotic diseases, and topics on nanoparticles, biosensors, microfluidics, machine learning, and other emerging diagnostic fields, plus 2 outdoor sessions. One was in the Philippine Genome Center and in Intramuros for the cultural event. So other pictures during the registration, during the scientific session, during the interactive session, and outdoor activities at the Philippine Genome Center and Intramuros. Now, for the outputs, the organizers were able to draft manuals, or a workshop manual, conference proceedings, a white paper and signed manifesto on how the participants can collaborate, primarily on how to improve diagnostics and vaccine designs in the UK and in the Philippines. And because of the limited resources, so as to maximize the impact of this worthwhile event, so through our media partners, portions of the workshop was broadcasted live to the industry stakeholders through social media and mainstream media in cable channel and in local station here in the Philippines. Now, other than that, so other contributions to sustainable development. So this event was a good tool or platform or avenue for some of the participants of another funded research projects, and also from the Newton Fund, but this time with the DA through the DA Biotech Program and the BAR, no, the UK, China, Philippines, Thailand Swine and Poultry Research Initiative. At the moment, there are 4 research teams from the Philippines, and out of the 4, 2 were able to use this platform or this workshop to be able to enhance further their research collaborations, update their results of their experiments, and plan on how to proceed with their studies. No? So one of those research team is our team with Dr. Bala of Brunel University. So right now we are working on a low-cost portable molecular diagnostic platform for rapid detection of poultry infectious pathogens, and funded by the Newton Fund and the DA Biotech. And in 2019, this was shortlisted as one of the finalists for the Newton Prize. And with the COVID pandemic, no, so our collaborators in the UK, they twitched the machine to be able to study or detect COVID in human specimens. Now, other than that, other contributions to sustainable development. So through the workshop, we were able to link Dr. Muhammad Munir and Dr. Jerry Kamer from UEP to make research on poultry vaccines using recombinant technologies. And as of the moment, they also have a PhD student also from the Philippines, no, working on this research. And there is ongoing collaboration to identify potential PhD students between UPLB and University of Liverpool. And the participants would like to use this workshop, and they wanted to propose for the establishment of the Philippine Society of Virology, wherein the attendees as the founding members, and also a proposal for the establishment of research focus groups on tick-borne diseases. Now, again, we would like to thank the British Council, the Newton Fund, DOST, through the Livestock Research Division of Picard, for funding this workshop, and also to our media partners, DOSTV, Pit Games, and Alagang Magaling, as well as the Philippine Genome Center for allowing us to visit their facility during the workshop. That's all, and thank you very, very much.
Thank you, Dr. Dennis. That was wonderful, and I enjoyed your sharing of the video as well, the highlights from the workshop, and to learn more about the work that you're doing. Also, you mentioned the swine and poultry work that's under Newton, and that's really great. We now invite our 3 speakers to join us for a brief question and answer session. And I welcome you to turn on your video, and also welcome our participants who are in this room. It's a small, cozy group, and I would like to open this virtual floor to our participants to ask any questions. You may put it on the chat, or if you feel like you like to speak, please raise your hands. So would anyone like to ask any questions? I know Danny may have one question, so I might pick on Danny to ask his question at this time.
Hi, thank you, Esther. I'm just interested first and foremost about the emphasis of Dr. Lisa Osorio's team in determining how gender has an impact to an initiative such as this Newton Fund project, and whether there is anything that maybe CEFAS or University of Cebu would like to continue following that. I don't know if it's a discovery or a realization, but is how do we sustain that kind of of understanding in terms of gender inequality?
Yeah, thanks, Danny. Well, our recommendations that I wasn't able to present was actually to be able to work together to further investigate the role of gender in seaweed farming communities in the Philippines. And as I said, this was based on our realization in the course of our study that, aquaculture seen through the lens of gender has not yet been fully studied. And we realized that in visiting these island communities where the aquaculture farms were located, we were assuming that this were mostly consisting of men who were farming, but then we saw that in seaweed farms there's actually a more um, evident workforce coming from the women. They were active in more of the farming tasks than men, but at the same time, men had a more decision making power on the farm. So, and this is also we found out that most of the respondents in our study, in the gender study that we initiated, was that the majority of the respondents were actually in the lowest income class. But later on, we know that there could be shift in decision making power if women gain more experience, they have more assets, so they have farm ownership was actually a form of empowerment, but there are still efforts needed to help them become empowered beyond just the domestic sphere. And so it's really very interesting question that we found out, and we wanted to pursue actually together with CFAS. And yeah, we have submitted a research proposal for grants to further our studies on this aspect. Unfortunately, we have not been quite successful to get more funding on this aspect. So we're still trying, but yes. Thank you so much for the question.
Yeah. We find this very much interesting because this complements the sort of priority being put to the gender equality by the ODA. And this we will learn maybe later when we do the final plenary on ODA. But in saying that, for example, us in the British Council, we put so much value in gender equality, not only in the composition of the teams, but also in the effect or in the impact of the any science work or research work that we do or we support, for example. So thank you, Dr. Osorio.
Thank you. If I may ask a question to Dr. Marissa Romero, because I was curious about what are some of the challenges you see in the science dissemination in the Philippines? What are some of the obstacles you face maybe in the science communications to the local public? And also a question to Dr. Dennis, if I may. I was wondering, how do you think the international partnership aspect of that research, especially the workshop you talked about for VetMed to come together, how has that enriched and added to the ecosystem in the Philippines? Thank you. Perhaps Dr. Marissa, please.
Yes, thank you for that question. Here at the PhilRise, we're very lucky to have a development communication division because, as you know, researchers usually tend to be very technical, and when we convey our messages, then the masses or the consumers or our stakeholders won't easily understand that. But that's just the way we are as researchers. But we're learning also, we're being trained also to popularize our language, so we can just like what I did, I tried to simplify my presentation as much as I could so that it's easier to understand, because research work can be, or scientific work can really be very technical. And even though we do details, but but I think the best thing that we can do is to just convey the highlights of our research to the community so that they will be able to understand it well. If there's scientific conferences, I think that's the time that we can be technical. But again, as I've said, with a collaboration with the development group and other partners, we will be able to communicate science clearly so that they will be very excited to gain the benefits from our research work. Thank you.
Thank you. Thank you, Dr. Marissa. And Dr. Dennis, please.
Thank you, Dr. Wong, for a very good question. Now, the 13 senior mentors from the United Kingdom, they were very helpful, no? And we are very lucky that they were able to find time in their very, very busy schedule to visit the Philippines and share their expertise. And more than that, we would like to emphasize that right now in the Philippines, we are at the molecular level in terms of diagnostics. But the arrival or the participation of the experts from the United Kingdom, they open new doors or new perspectives on how diagnostics could be by integrating machine learning, nanotechnology, and other emerging fields, especially biosensors in livestock and poultry. So again, we are very grateful for this opportunity. And again, thank you for the support.
Thank you. Thank you both for answering. I wonder if anyone else have questions.
Hello.
Hi. Yes, Crystal. Please go ahead and state your question.
Actually, ma'am, this is not a question for the British Council itself regarding Newton Agham. Currently, ma'am, I'm a master student here in UK, and how can I apply or engage with the Newton Agham so I could like fund my dissertation there in the Philippines? Like, should I connect with university or research institution in the Philippines, or should I apply individually using recommendations from my university here in UK?
Hi, Crystal.
Yes.
Right. For the coordination on the PhD scholarships, first, for this year, we are not running any programs just yet. As well, we are following the rhythm and also the decisions being made by our funders, which is the UK base. Nonetheless, the British Council also offers other programs that could support your research, which we can coordinate separately. You may email me, and then we can have a conversation, the both of us, in on how the British Council could support, or maybe lead you with organizations who could also support, depending actually on your research agenda.
Yes. Yes,
Thank
sir.
you, Krista.
You can
I
message
will,
me your email address. Thank you.
sure. Yes.
Thank you.
All right. Thank you.
And I think with that, we are out of time for our Q&A. And I thank again our three speakers. And I welcome all of us. If you have other questions, you can contact them. And I'll pass it back to Danny.
Thank you, Esther. Before we leave, I'd like to ask if you'd like to open your turn your cameras on so that we can have a photo opportunity within our group, which likely the other groups are also already doing at by this time. So if you would like to, and if you would, please turn your cameras on so we can take some photo opportunity specifically for group C. All right, so please hold on to your smiles while our staff, designated staff, tries to take screenshots. Chris Ann?
Okay, we're good. Thank you.
All right, thank you. Thank you everyone for joining. Now, I'm going to share my screen about returning to the room. But if you can see at the bottom right of your room, you can see the blue button that says leave room. This will lead you to back to the main room. And then I think we are yet to wait for other for the health session to complete their group presentation. So while we're waiting, we can have a short bathroom break, and then return finally for the third session, which will be delivered by the Undersecretary Guevara, Dr. Leia Buendia, and also Dr. Clarissa Domingo. Again, lower right of your lower right of your screens. I'm
Thank
going to
you.
stay here and remain here just in case. But I'd like to thank Esther Wong for being the moderator this afternoon
Thank
for
you,
session
Danny.
2.
Thank you, everyone. Hope you enjoyed the sessions. See you back in the main room.